I'm very, very excited to be here. So I don't know how much time I have or how much time you have to be with me, but I'm going to try to walk you through a little bit what we're doing. So the title of my talk is Olfactory Dysfunction Neurodegeneration Towards an Early Detection. So what we are trying to do is to find two diseases, Alzheimer's disease and Parkinson's disease, which are both associated with a reduced sense of smell very early in the development of the diseases. And we try to see whether we can use this information either for early detection or for better understanding what's going on with these diseases. So there's different neurodegenerative diseases. You may have heard of most of them. So Alzheimer's, Parkinson's, multiple sclerosis, Lewy body dementia, frontotemporal dementia, Huntington's disease, ALS, and so on. And so there's other neurodegenerative diseases. And what's the particular case about that is that there is a degeneration of neurons in the brain, and this is progressive. So they get worse and worse. And at some point, this will have clinical consequences. And at some point, this will then lead to the death of the person. There's very different underlying causes that could lead to the neurodegeneration. In some of these diseases, in many of these diseases, you have a aggregation of pathological proteins in the brain. So the proteins accumulate, they cannot be taken away, and they lead to the degeneration of the neurons in specific areas that is disease dependent, and eventually lead then to the breakdown of the brain and to the death of the patient. In order to develop therapies for these diseases, we have to detect them very early. So we don't have to detect them when a large chunk of the brain is already destroyed. We try to detect them very early in the development of the disease, and we think that olfaction can help us in this. And so here I have some number. This is not from a scientific publication. I just tried to aggregate the cases for Germany so that you get an idea. 80 million of people in Germany. How many people suffer from Alzheimer's, Parkinson's, and so on. I group them together like this because this is how I found them. But there is evidence that, for example, Lewy body dementia and Parkinson's disease are two expressions of the same underlying pathological phenomenon. And I'm going to talk about this. And as I said, Alzheimer's and Parkinson's are characterized by an early olfactory decline. So this is interesting because this olfactory decline, we see it earlier than the manifestation of the typical symptoms that you think about. When you think about Parkinson's, you have the motor symptoms, the shaking. When you think about Alzheimer's, you have the cognitive, the dementia, Alzheimer's dementia. But earlier, before even the appearance of these symptoms, we have a reduced olfactory function. So here, what I'm going to talk about, first I'm going to talk about Parkinson's and Alzheimer's disease. I don't know to what extent you're familiar with these diseases. I'm going to talk about it. If you say, okay, we know all this, give me a sign. And then I will jump on. Then I will talk briefly about the sense of smell, because I also assume that most of you, not all, most of you are not so familiar with the sense of smell. So we'll talk a little bit about the sense of smell, then about the olfactory dysfunction, neurodegeneration, and then I'm going to dig a little bit deeper into the case of Parkinson's disease, because we have made a series of studies. An outlook, then I'm going to talk a little bit about Alzheimer's disease, but when I come to Alzheimer's disease, you probably are going to be tired, and so this is very short. And but if you want me to talk about more about Alzheimer's disease, well, you have to invite me back, and I have to talk about that. So, what are Parkinson's and Alzheimer's disease? Parkinson's disease has first been described by a English physician, James Parkinson, in 1817. It's a progressive degeneration, so neurons in the central nervous system that degenerate. And in his first description, he called it a shaking palsy. What we typically think about when we have Parkinson is the person that shakes, that trembles like this. You also see people that suffer from Parkinson's on the street. They have a very typical gait, a typical way to walk. They walk, they don't shake they don't swing their arms. Their face is like a mask. They make very little steps like this. This is the so-called Parkinson shuffle. It's something very, very typical. What you see on this image is the change in body posture that goes from the normal kind to the very advanced kind of Parkinson's. Then you have shaking maybe. So this is something that you see even on the street. The underlying cause of the disease, or the underlying pathomechanism, is that there is a misfolded protein, the alpha-synuclein, that, because it's misfolded, aggregates, cannot be taken away, forms those Lewy bodies within the neurons, and the neurons then degenerate. We do not know what causes the aggregation of these proteins. There's different hypotheses what may cause the disease, but we don't know yet. It may be some agent that comes from the outside into the brain, and we can talk about that after my presentation here. What is typical for the disease is that there is a dysregulation of the dopaminergic system. The dopaminergic neurons of the substantia nigra in the brainstem degenerate, and they play a role in the control over motor function. They degenerate, and then you get the symptoms, the motor symptoms of Parkinson's disease, which is the involuntary shaking, but also the difficulty to start moving. So they have a reduction of voluntary movements. It's difficult for Parkinson's patients to start a movement. They don't have these involuntary movements. The face has no facial expression because the muscles are like don't move, and all this is caused by this degeneration of the dopaminergic system. It affects approximately 1% of the 60 years plus patients, and 4% 80 years and plus. And right now we have only symptomatic therapies available. There is no therapy that allows us to cure the disease, also because we detect the disease too late, when a huge chunk of these neurons is already degenerate. If we look at Alzheimer's disease, that was first described by a German, Alois Alzheimer, in 1901. It's equally a progressive degenerative disorder of the central nervous system, but it affects cognition and memory in the first place. So it's another system in the brain. And again, it's the underlying pathomechanism is the aggregation of proteins. It's different proteins. It's a protein tau that causes these neurofibrillary tangles within the neuron and amyloid plaques outside of the neuron. And this causes the neurons to degenerate, to die, and the disease is characterized by lesions to the cortex that now are not in the brainstem as with Parkinson's disease, but they're in a different area of the brain, in the temporal cortex, which is the seat of memory, acquisition of new memories, and retrieval of memories. And therefore, the consequences of this are that people get memory problems, they get dementia eventually. It is more common. This patient here is the first patient described by Alois Alzheimer that died very fast. It was a relatively young patient that died very fast. And when he looked at the brain, he saw these tangles. And what we see nowadays with Alzheimer's disease is not so much people in their 40s and 50s, but more people in their 60s and 70s that suffer from dementia. So it affects 2% of 65 years and plus, and 20% of 80 years plus. So it's a disease that is very important in an aging society as in the Western world. Only relatively recently, there were some curative therapies that were introduced, where you take some very expensive medications that would help us to take off these tangles, to reduce the load of protein tau and amyloid plaques. But even these interventions, although they seem to be curative, they have only limited effects, because they kind of slow down the progression of the disease. They get dementia too, but they get the dementia a little bit later than the ones that don't take. So we're relatively far away from this. What you can see on the next image, if it wants. Yeah, here you see, this is a cut through the brainstem. The brainstem is approximately here, you cut through. So here we are on the level of the mesencephalon, and what you can see here, this is the brain of a healthy, also died, but didn't die of Parkinson's disease. And you see here, did I do this by touching the screen? That's very exciting. So what you can see here, the black is the substantia nigra, where you have these dopaminergic neurons. They contain some iron, and the iron makes them black, appear black when you look at them. And when they degenerate, you see something like the brain on the right, where you do not see this this accumulation of black substance. So the substantia nigra becomes gray and you cannot see it. And what you have in Parkinson's disease is this accumulation of the alpha-synuclein that forms these Lewy bodies in the neuron. No. And what you can see here.
I think it's just lagging.
Yeah, why? Here. This is the typical Alzheimer. This is a regular brain, and this is a brain of a patient with Alzheimer's disease. And what you see in the ventricular system, especially in this area of the temporal lobe, you have the hippocampus, which is the region that's responsible. Thank you. Responsible for the for memory retrieval and formation. And these are the regions of the brain that show the first degeneration. And then with progression of the disease, you have a shrinkage of the whole cerebral cortex, because in both cases, the disease spreads beyond the centers of the brain that are touched in the first place. And then you have a generalized neurodegeneration. And also with Parkinson's disease, eventually you will have dementia. Here you see the disease progression of Parkinson's disease. In color, you see the areas of the brain that are touched first. And what you see in the premotor phase of the disease stage, so this is before a diagnosis is made, you see that two areas of the brain that are touched as first, where you have the accumulation of those proteins, which is the olfactory bulb. It's approximately here, and the vagus nerve that is here. So that gives us an information, maybe whatever causes disease enters the brain via these pathways. In these stages of disease, you have very unspecific symptoms, such as olfactory dysfunction, as I already said, maybe some constipation, but a lot of people have constipation. And only later, when the motor symptoms appear, so you have the neurodegeneration of the substantia nigra, then you get the diagnosis. This is typically also a time where people have sleep disturbances. We will talk about that a little bit later. And then later on, it spreads, as I said, and then you have also emotional and cognitive dysfunction. So this disease does not stop with the motor symptoms, but goes beyond. And if you ever go to a retirement home where there's patients with dementia, often they have underlying Parkinson's symptomatic as well. So it's not because somebody has dementia, it's Alzheimer's. You have different causes for dementia. It can be vascular. It can be Alzheimer's, it's the most common, but it can be also a Parkinson's cause. When we look at Alzheimer's disease, the progression of the Alzheimer's disease, it starts in a different area of the brain. It starts in the temporal cortex, typically with the subjective cognitive decline. These are people that say, I feel like I can remember less things, but they are still in the normal range when they test them. Then you have a little bit later a memory loss. You have a mild cognitive impairment, so they have a slight reduction when you test them, but it's still not pathological. Later on, it will become a manifest memory loss. They have an emotional disbalance, as you can see, for example, with the president of the United States that has an emotional disbalance. Maybe he has some form of neurodegeneration, but I'm not here to speculate about that. And then later on, you see how the disease spreads all over the brain, and then it comes to a loss of autonomy. People cannot take care of themselves anymore. They cannot go to the bathroom. They cannot wash themselves. They become at some point just very dependent on help. And if they get the right help, they can still live for quite a long time, which means that for the society, it becomes very expensive. It becomes difficult for the relatives to take care of them, and it becomes expensive because these people need a lot of care. So this is another reason why it's not just, of course, we not think about money only, but we also want to help the people to live healthy longer. And so this is the idea why we need to do something. Do you have any questions with regards to this? If not, we will. Yeah.
So in the Parkinson progression pictures, the cerebellum was not marked. So I was wondering if that's just left out.
The cerebellum is not typically involved in the disease progression. I do not know if this is just because people don't look at it, but it's not the typical cerebellar symptomatic that the people have. If you have cerebellum dysfunction, the movement. It would affect movement, but the movement is affected in a very different way. So if somebody has a cerebellar ataxia, for example, the way they express movements is like somebody who's drunk. So they have a problem with coordination, they have a problem with balance and so on, but it's not what you see with Parkinson's. They don't fall over in Parkinson's disease.
So if I may add anything here, so the motor deficits in case of Parkinson's disease is caused by the loss of dopaminergic neurons within the striatum. So the D1, D2 pathway in striatum, they are dopaminergic cells. They're managing the especially initiation movements, and it's a bit of a different pattern of motor deficit compared to the cerebellum.
Thank you. Okay, so now I'm going to talk about the sense of smell a little bit. We can with the sense of smell, we can perceive the composition of our environment, not just the outside environment, also our inside environment, what we have in our mouth. We perceive it with our sense of smell. We can smell different volatiles, and we can distinguish billions of odors. It's an extremely large number of odors. There's a paper that said 1 trillion odors. Maybe it's not 1 trillion. This paper has been criticized. Maybe it's just 100 billion, maybe it's just 10 billion, but it's an extremely large number of odors that we can distinguish, much more than actual substances, volatile substances, because if you mix two substances together, they will have an odor that is different than the individual ones. When we smell something, here's a little cut through the skull. Here you have the brain. Here you have the nasal cavity. This is the nose. These are the nostrils. These are the lips, the teeth, the tongue. Here you have the oral cavity. Here you have the nasal cavity. And the smell perception takes place via an interaction of the volatile substances that come into our nose, either from this side, this is orthonasal olfaction, or from the back side. It's funny. They can also come from the mouth. If you have something in your mouth, you close your mouth, but you can still perceive what's going on. And up here, where you have those yellow fibers, you find the olfactory epithelium, neurons that have receptors that can interact with these volatile substances and then send a signal to the brain. And the first structure in the brain that receives the input from the olfactory epithelium is the olfactory bulb that is situated right here at the beginning. So those fibers, in their ensemble, they form the olfactory nerve. The fibers go through the skull, and so the skull has many different little holes, and then end in the olfactory bulb that is situated right here between the eyes, a little bit above the eyes. And then the signal is processed and the information travels further on. We can smell smells or odors. Odorants can enter the nasal cavity via two different pathways. The first one is what you typically would think when you think about smelling. So let's say you have a beautiful smoky Earl Grey tea in your glass, and you sniff it like this. Well, you suck the air into your lungs, and on the way there, it goes through the nasal cavity and reaches the olfactory epithelium in this upper portion of the nasal cavity. When you have the liquid in your mouth, you close your mouth, your lips, but the olfactory molecules can still enter the nasal cavity via the back door here. This is called retronasal olfaction. It plays the most important role in flavor perception if you eat something. So if you lose your sense of smell, the one thing that you notice is that things don't taste good anymore. But it's not the taste, it's not the fact that sweet, sour, salty, you can still perceive. What you cannot perceive is the aroma of this by this retronasal olfaction. So there is the sense of smell can not work, and it's actually something that is quite common. Olfactory dysfunction, we call this. We have two different kinds of olfactory dysfunction. We have quantitative olfactory dysfunction and qualitative. Quantitative means you smell less. You can also smell more, but I'm not going to talk about that. So if you smell nothing at all, it's called anosmia, complete loss of olfactory function. If you smell less, but not completely nothing, then you talk about the hyposmia. These are quantitative olfactory dysfunctions. And then we have qualitative olfactory dysfunction is when you smell things, but they are altered. They don't smell as they're supposed to smell. For example, vanilla doesn't smell like vanilla anymore, but smells like burnt rubber. Okay, this is called a parosmia. There's also phantosmia, is when you smell odors that are not around, but that's relatively rare. I had parosmia when I had COVID for the first time, and this was triggered by vinegar. So when I ate vinegar that my lovely mother had prepared, a salad with the vinaigrette that my mother had prepared for me when I was sick, I thought, oh my goodness, something went bad here. It smelled like moldy, mushroomy. And then I found out, well, it's not the food that my mother prepared, but it's actually me, because I smelled the vinegar as this strange odor. And when I recovered from COVID, this went on for several weeks and was not just limited to vinegar, but it was also triggered by red wine. And I was living in Italy back then, where you drink red wine like 3 times a day. And so this was quite problematic, but luckily I got rid of this parosmia. So you see here that approximately 20% of the population suffer from smell dysfunction. They have some kind of either anosmia, hyposmia, parosmia, combinations thereof. And there's different diseases that can cause olfactory dysfunction. I just talked about COVID. This is quite common, but people with chronic rhinosinusitis, viral infections, it's not just limited to COVID, also the flu and other viruses can cause it. Traumatic brain injury, like a concussion, can cause olfactory dysfunction. There's people that lose their sense of smell, we don't know why. And there are the neurodegenerative diseases such as Parkinson's and Alzheimer's. They can all cause olfactory dysfunction. So you can already imagine, if you want to use olfactory testing as a marker for neurodegeneration, you have to make sure that you don't include these kind of cases here, which are quite common in the population. So what about olfactory dysfunction in neurodegeneration? I did this study 2 years ago, was published last year, where we investigated a cohort of people living in a valley of northern Italy, of Südtirol. And in this cohort study, they invited all the adults of this valley. Approximately 80% of the adults participated, and of those 80%, approximately 6,000 also underwent olfactory testing. So we had the olfactory testing data and odor identification tests, 16-item odor identification tests, and we had a lot of information about these people. We have genetic data, we have the microbiome, we have demographic data, and so on, and medical history. And so we started looking into what are the causes, which factors determine if they have a good sense of smell or a bad sense of smell. And I report some of these here. We had many more factors that came out significantly, but the most important ones, I mentioned them here. So we have age, sex, and education. So the younger people perform better than the older people. It's something that was known before. Sex here, females perform better than males on average. It's not a very huge effect. There's a lot of variation, but on average, females outperform males. Why this is, we don't know. We can discuss about that. Education, people that have more years of education score better in these kind of tests. There's an odor identification test. So in order to know, for example, cilantro, you have to be exposed to cilantro. And we think that there is some association between years of education and exposure to certain odors and odor descriptors. But this is not what I want to talk about here. We also have lifestyle factors, smoking, alcohol consumption. Smoking, if you stop smoking, I don't know if anybody still smokes, but if you stop smoking, your sense of smell gets better. And that's good news. So it would be a good idea to stop smoking. Alcohol consumption is a little bit more interesting. People who do not drink at all have worse scores than people that drink moderately. So a little bit of alcohol seems to help for the sense of smell. Why? There is different interpretations that you would have to evaluate. One of them is that when you drink alcohol, typically there's flavors involved, and you may train your sense of smell. But there were 3 medical conditions that were very interesting. So people with a history of a skull injury, a traumatic brain injury, had reduced and had lower scores. And people that scored high on a Parkinson screening test, and people that scored low on a dementia screening test, which both means that they go towards Parkinson's or they go towards dementia. So this is one example that in the overall population, you have these effects. You can observe them. I'm going to show you a result here. Dr. Schöpf, this is the moment where you have to take a photo. So we did this. It's a well-established fact that both Alzheimer's and Parkinson's disease are associated with reduced olfactory function. We did a meta-analysis with Shadi Rahariel, who was here early this year, back in 2012, where we looked at the time, it was about 70, 80 publications on Parkinson's and Alzheimer's, and we looked what is the effect size that those two diseases have on olfactory function. And we looked at different olfactory tasks. We looked at odor identification, which basically is I give you an odor and you tell me what it smells like. We looked at odor discrimination. I give you two odors and you tell me are they the same or different, or I give you three odors, two are the same, one is different, you tell me which one is different. We did odor detection, which is basically I give you odor in different concentration. We checked from which concentration are you able to detect it. And we do odor recognition, where I give you an odor and at some interval later, we give you the odor again and you tell me, have you already smelled this odor? Yes. So the odor identification test is the most commonly used. The other two here are also relatively commonly used. Odor recognition tests are less commonly used. But what I want to show you, what I want to stress here is that Alzheimer's disease has the dark gray bars and Parkinson's disease, the light gray bars. Here you see the effect size. You see both diseases have a very huge effect on all olfactory, different olfactory tasks. But when you look into more detail, you see, for example, that Parkinson's disease affects those different tasks in a relatively homogeneous way. Whereas Alzheimer's disease has a very huge effect on odor identification and odor recognition. So tasks where you have a memory involvement, whereas odor detection has a relatively limited memory involvement, is less effective. So this shows us, while both diseases affect the sense of smell very strongly, but they may not affect them in the same way. And perhaps there is something like a olfactory impairment pattern, so that the diseases affect the sense of smell in a very specific way, in an Alzheimer's specific way, in a Parkinson's specific way, and they may be different from other diseases that cause olfactory dysfunction. So this may be a way to establish a disease-specific olfactory impairment pattern. And this is where I want to go from now on, because if we want to detect Alzheimer's and Parkinson's disease early, well, we have to, it's not enough to just test of odor identification. We have to look at different tasks, and then we see a pattern, and this pattern will allow us to say, well, it's rather Parkinson's or it's rather Alzheimer's, or it's maybe chronic rhinosinusitis. I already stressed this before. Olfaction is affected very early in Parkinson's and Alzheimer's disease. So what you see here in red, in green, rather, sorry if there's any colorblind person here, did not think about that. So what you see here in the green line, the top line, is the decline of motor function for Parkinson's or cognitive function for Alzheimer's. So the people live, this is the age they have, and at some point they have a decline of this, either motor or cognitive function. At some point they go to the doctor and they get the diagnosis. This is the diagnosis here. And then the function declines further. When we look at olfactory function, we see that the decline of olfactory function starts already 10 to 15 years earlier. And once they reach a diagnosis, the olfactory dysfunction is relatively well established and does not change a lot anymore. But this difference here is the window that we would gain if we could detect the diseases earlier based on olfactory function. And I'm going to show you how we hypothesized that we could do that. It's a study that I participated in a lifetime ago, basically, for some of you, maybe. So this was done in a clinic that focuses on olfactory dysfunction. Okay, so it's a clinic in Germany, in Dresden, where people go when they lose their sense of smell. There's not a lot of specialists, and there were even less so in 2004. So the patients go there and they get evaluated, and then depending on the underlying cause, they get a treatment or not. So if they have a chronic rhinosinusitis, they get either some spray or something for their nose, for the mucosa, and they get that. But there is one group of patients that have idiopathic olfactory dysfunction, which means that we don't know what caused the olfactory dysfunction. They didn't have a concussion. Their nose was free. They didn't have a history of a infection or something like that. So we don't know what causes it. And these are the ones that are probably the ones that could develop a neurodegenerative disease. So we took these 30 patients. We sent them to the neurologist. So they had smell problems, nothing else. And when the neurologist evaluated them, they were normal. So 0 of 30 patients had pathological neurological symptoms. But then the neurologist did a second exam where they really focused on motor, neurological motor evaluation. So much more focused on that than the regular neurological evaluation. And 4 of 30 patients had somehow a little symptoms that may be borderline pathological. Then we sent all these 30 patients to do an ultrasound of the brainstem. With the ultrasound of the brainstem, you can see here the echogenicity of the substantia nigra. So you see how healthy the substantia nigra is. And 11 of the 30 patients had some borderline pathological symptoms here or results here. And then we invited these 11 patients to do a SPECT imaging where we can see the function of the substantia nigra. Only 1 patient did not want to participate in that, and so we had 10. And of those 10, 5 had pathological symptoms or results in this evaluation. This is somehow summarized here. So these are all the patients. These are the ones that had the neurological, darker gray means pathological. So these are the ones that had a pathological evaluation in the neurological evaluation. Then we did the TCS. They were normal. These were pathological. Then we did the SPECT, and you see the results here. So what this means, we have 30 patients, and of those 30 patients, 5 have some pathological results in an ultrasound and in a SPECT imaging. So these are candidates for later developing Parkinson's disease. There was a follow-up study, and of those patients, 2 indeed developed Parkinson's. So that may sound exciting, but on the other hand, it also means that you have 30 patients that you worry, and 28 of them do not get Parkinson's. So we have to get much better. But the basic idea how we would proceed is this. We would detect somebody based on their olfactory function, and we could guide them towards a diagnosis. And if we have that, then we have a window where we could intervene if we have therapies. We don't have them yet, but we could start developing therapies. If I say we, it's not me, it's not you, it's other labs that work on therapies, but this is the idea. So now I'm going to talk to you a little bit about one way that we think that this could work. So to summarize, we try to find a disease-specific pattern of impairment. And if we detect this disease-specific pattern, then we could apply this to individuals that are at risk and see, do they express this pattern? And if yes, then we could start to intervene. So here I'm going to tell you a story about our travel into trying to determine, can we find a disease-specific pattern? And so we think one aspect that we could look at is the trigeminal system. So now you think maybe, what is he talking about? What is this, the trigeminal system? The trigeminal system is a third chemosensory system next to smell and taste that allows us to perceive the spiciness of hot peppers, the freshness of mint, peppermint, and so on. So all these spices and herbs that I present here, they activate the trigeminal system. It is linked to cranial nerve number 5, so it has fibers in the nose and in the mouth. And it allows us, when we eat something, to perceive, oh, this is spicy, this is burning, this is tingling, this is stinging, and so on. It's a chemosensory system because it's also responding to the presence of chemosensory stimuli, of chemical substances that we have in spices and herbs. But it's unrelated to smell and taste. So it's a third chemosensory system. And we did, again, very long time, a study, that's me, in younger days. Usually people that lose their sense of smell also get a reduction of their trigeminal sensitivity. So people that lose their sense of smell, cannot smell anymore, they also get less sensitive to spiciness and freshness. And one way to test the trigeminal system is with this device here, where we present a trigeminal stimulus to one nostril, eucalyptus, for example. We blow eucalyptus into one nostril and we blow air into the other nostril. And we ask the participant, air that doesn't contain eucalyptus, we ask them which side has been stimulated, and they respond this to us. So you have a 50% chance. So we repeat that a lot, so 40 times, then we count how often were they able to correctly identify the site that has been stimulated. If we use a substance that just activates the olfactory system, we are not able to do that. Like, for example, phenylethanol is a rose odor. If we do that with phenylethanol, which does not activate the trigeminal system, we are at chance. We don't know if it's the left or right nostril that has been. But if you use eucalyptus or menthol or something else, then we can do it. And what you see here is the result, again, a study that we did in 2004, in patients with olfactory dysfunction in gray, and healthy controls in black. We used eucalyptol, what I just talked about, so it smells like eucalyptus and has this fresh component, or benzaldehyde that smells like almonds and burns a little bit in the nose. And we did it 40 times, and what you can see here is that the patients that have lost their sense of smell have significantly lower trigeminal abilities. So there's a significant difference. They're more or less at 20, which is a chance level, because you have a 50% chance to report that correctly. So this is a measure of the trigeminal system. And there was an old paper that suggests that they investigated Parkinson's patients, and they had said, well, we did not see, they had normal responses to a trigeminal stimulus. So we took this paper and dug a little bit deeper. So that's Cecilia that you know, and if you're lucky enough to ever meet her, I invite you to do so, because she's a very brilliant researcher, just is finishing her postdoc. And so we wanted to compare Parkinson's patients with controls, but we also wanted to include individuals that have lost their sense of smell due to another cause, because we want to find a disease-specific pattern. So we cannot just compare them with healthy controls. We know that they have a reduced sense of smell. We want to compare them to people that lose their sense of smell due to other causes. So we tested olfactory sensitivity. So you see here Parkinson's PD and NPOD's non-Parkinsonian olfactory dysfunction. So if we look at the sense of smell in controls, they score high here. If you look at Parkinson's patients and patients who have lost their sense of smell, they're both significantly reduced, but there's no difference between them. And then we did a test for trigeminal sensitivity. We compared the controls with the patients who have lost their sense of smell due to another cause, and they have a reduced function. But if you also looked at Parkinson's patients, we see that the Parkinson's patients do not have this reduction. They rather have an increase, if anything. So this may be this specific disease-specific pattern, which means that in Parkinson's disease, you have a reduction of the sense of smell, but you do not have the reduction in trigeminal sensitivity that you usually have. So then we looked a little bit more into detail. This is a study, again, by Cecilia, where we gave our participants 8 different odors, and we asked them to evaluate them on olfactory dimensions that you see up here: pleasantness, edibility, familiarity, and 3 trigeminal dimensions: coolness, warmth, and piquancy. And I don't know if you can see it, but you see the dark black is the patients, the gray is the people with normal sense of smell, and you see that for the olfactory dimensions, you have a huge difference between both groups, but not for the trigeminal dimensions. I think I have. So here you see familiarity, and we'll have another one. Well, okay, but you get the point. So then we started looking into the pathway, the trigeminal pathway from the nose to the brain. Where are the changes happening? And so we did this negative mucosal potential, which is basically putting an electrode into the nasal cavity and see the responsiveness of the nasal mucosa to trigeminal stimulations. And what we see here is, again, I don't want to go too much into detail into this, but we see again that Parkinson's patients and non-Parkinsonian and patients with non-Parkinsonian olfactory dysfunction also exhibit a difference in this peripheral measure. Then we looked at the brain with EEG, and again, we saw that there are differences between Parkinson's patients and patients with non-Parkinsonian olfactory dysfunction. So it seems like that we are somehow finding this specific pattern on different levels. We saw it behaviorally, we see it with electrophysiological, peripheral, and central measures, and we even see it here with, this is a MRI study where we looked into, oh God, this is very bad resolution, but basically what we show here is, again, we can talk about this in details, but for the time being, just what I want you to show here. If the participants did a odor identification task, which is a smell, an olfactory task, we saw that the modularity distinguished Parkinson's patients from the other two groups. And then when they did an odor localization task, which is a trigeminal task, when you say left or right nostril, again, we could separate the Parkinson's patients from the other two from the other two groups, which are healthy controls and people with non-Parkinsonian olfactory dysfunction. It's not completely surprising that you see these differences because, remember, Parkinson's patients, they have a sick brain, whereas the other ones, they have a healthy brain. And so it's not completely surprising that this sticks out. But what this tells us that on every level of processing, we can see a distinction between Parkinson's patients and patients who have lost their sense of smell due to another cause. And we suggested this model, and again, we can talk about this in more detail, but here is how the olfactory and the trigeminal system interact in the periphery on the level of the olfactory bulb in the brain. And when you lose your sense of smell, for example, due to chronic rhinosinusitis or viral infection, you have an impact here on the level of the olfactory mucosa in the periphery, which then triggers different compensatory mechanisms that result in something that is that accords to what we show. Whereas in Parkinson's disease, it's probably not the periphery, but it's the olfactory bulb that is affected, because it's an area of the brain that is very early affected, and that then triggers other mechanisms, and these are the ones that we observe. And so now we're still carrying out series of studies where we try to test this model. We did another study, and this is one where Jie was involved, where we wanted to see, if we look at the olfactory bulb here, do we see differences between Parkinson's patients and non-Parkinsonian olfactory dysfunction patients, the ones that we that are interesting. If we just look at the size of the olfactory bulb, we can measure it with an MRI at T2. We see that all patients that lose their sense of smell have a smaller olfactory bulb compared to controls. But we don't see a difference between those two. But then we used those advanced abilities of Jie, and we looked into the areas surrounding the olfactory bulb. And if we looked into that, so this is probably not the olfactory bulb per se, but it's the areas around the olfactory bulb, orbitofrontal cortex above, where we have effects and so on. We see that those areas that you see highlighted here in red, these are the areas that allow us to distinguish with a relatively high accuracy between those two patient groups. Again, on the pathway in identifying a disease-specific pattern. We also did this with structural MRI data, where we looked not just at the olfactory bulb, but we looked at the overall brain areas. And again, we were able to distinguish between NPOD, non-Parkinsonian olfactory dysfunction, and Parkinson's patients on a image-by-image level with a relatively high accuracy. That, again, is something quite interesting. So depending on what level we look at, we see these differences between those two groups. And this is something that we're quite interested in. We then also looked into white matter integrity that connect these different areas here. There, we did not really see a difference, but probably we have the sample size is too small. These studies are relatively expensive to carry out because you need to get the participants, which is not very easy. And then you have to convince them to go into a scanner. They should not move too much, which is problematic with Parkinson's patients and so on. So it's not very trivial to do these kind of studies, but we also looked into the white matter integrity. We saw some effects, but they are not straightforward. So what are the next steps? Well, this is my friend Basil, that is a ENT doctor with this device that we use for trigeminal testing. We would like to develop a method that is faster to assess trigeminal sensitivity. Right now, we do this very, very crude method to test the trigeminal system. It takes us an hour. We get a number between 20 and 40, which is really not good. So we are looking into, are there different ways that we could get the information faster? We're working on it, but again, it's technically a little bit challenging. We think that the way to go is to vary the duration of the stimulus But again, it's not straightforward. And another idea, of course, is this should be something like okay, well, so we want to look at individuals that are at risk to develop Parkinson's disease. And we would like to know, if we have somebody who's at risk to develop Parkinson's disease, do we see already this disease-specific pattern? If yes, well, does this give us any prediction about if or when they would convert to neurodegeneration? So one important population that is at risk are individuals with REM sleep behavioral disorder, which is a patient population Shadi works on. So I don't know if you're familiar with it. I'm going to summarize it briefly. People, usually when we sleep, we don't move, right? We have different sleep stages. There is the REM the REM sleep stage, which is rapid eye movement stage, which is the stage of sleeping when we dream. And it's called REM because it's rapid eye movement. So we move our eyes, and this means that we know that somebody's dreaming, but the rest of the body is paralyzed. Okay, usually it's just the eyes that move. The eyes are closed, so you don't even see it, but the body is paralyzed. There's some individuals that develop this REM sleep behavioral disorder where they start moving. They live their dreams, but it's not a good thing. You know, you lived your dream. You dream that you're running away. Well, you move your feet. You dream that you're, in a fight with somebody and you beat up your partner next to you. So this happens. This can be caused by medication and so on. But there's a large group of individuals that have an idiopathic RBD, REM sleep behavioral disorder. So this means that there is no underlying cause. And these individuals are at very high risk to develop Parkinson's disease or Lewy body disease, which is related to Parkinson's disease. So what we want to do is to test these individuals and see, do we see this disease-specific pattern? And if yes, does it allow us to predict when they would be converting? It's not that easy as it sounds. In Montreal, there's a huge cohort of these patients. So in theory, we could test them, but the researchers are very, very jealous about their patients. Are very solicited a lot. So they do not want them to participate in too many studies because then they stop participating in the original cohort study. So they tell me something like, you have 5 minutes with them. You can have them for 5 minutes. But our trigeminal test already takes 30 minutes. And if you want to do EEG or NMP or something like that, it's going to take us 4 hours with those patients. And we don't have that. So that's a challenge that we're working on, to find a way to get our measurements done much faster. Another aspect that we're looking into right now is, I talked briefly about orthonasal and retronasal olfaction. So if you have a problem with your sense of smell, you also perceive the food less in your mouth, and that may have an implication on what you eat and how you eat. So we are currently looking into comparing orthonasal and retronasal olfaction between Parkinson's patients and patients who have lost their sense of smell due to other causes. We look into flavor perception, and this may be tell us something about the disease and maybe help us for prediction, but it may also tell us something about quality of life of these patients. What can we do to improve their quality of life? So that's another aspect that we're looking into. And we are currently trying to use a new method that has been developed at the Karolinska Institute, where we measure the activity of the olfactory bulb. It's a very new measure. There's now 4 or 5 papers out where they have done it. And that is interesting because it's an intermediate step between the mucosa and the central nervous regions that we can evaluate with this new method. And we want to look into again, comparing Parkinson's patients with people who have lost their sense of smell due to other causes. And then, of course, it would be very interesting to pursue a little bit what we have started doing with Jie in 2021, where we did this. She did. I just put my name on it. But we looked into different methods using machine learning for the identification of Parkinson's disease. And you can see here voice, gait, handwriting, MRI, SPECT, PET, and other ones. So these are all other methods. They have nothing to do with smelling. And we think that if we can integrate all these different methods together, then we would be able to reach a much higher probability to identify correctly who has the Parkinson specific pattern, who has not, and that will allow us to get much closer to our goal, which is to eventually identify these patients much earlier. I'm going to use my last 5 minutes to briefly talk about Alzheimer's disease. So no worries, I'm not going to talk as long about Alzheimer's as I did with Parkinson's. But it's just to give you a summary of studies that we have done in the last 5 years. So basically since the beginning of the pandemic, we know that Alzheimer's disease starts with a very unspecific stage, which is called subjective cognitive decline. Again, these are people that feel that their cognitive function has been reduced. So this is something I'm aging, I'm 51. You know, you're not as smart and you forget things and so on. But this goes beyond that. This goes beyond the normal aging. So it's very fuzzily defined. But we're looking into individuals with this SCD because they have a higher probability to convert to MCI, mild cognitive impairment, where you see. So if you test in someone with SCD, do memory tests, they have completely normal results. The only thing that is different is that subjectively they feel it's not as sharp as before. But they have a higher probability to go towards mild cognitive impairment, where you actually see a reduction in test. And those have a higher probability to go to the dementia state, where they're in the pathological range. So we want to see, is there an association between olfactory function and memory function in healthy people, in individuals with subjective cognitive decline, and so on. So we did a meta-analysis. This was what we could do during the pandemic. And you see that even in the individuals with subjective cognitive decline, they have a reduction in their olfactory abilities. We looked into, these are healthy people, healthy elderly people. Is there an association between olfactory function measured with identification tests and with threshold tests and different forms of memory? And we found here delayed recall. We found an association, immediate recall, global effects for both odor identification and also for odor detection. We did a study. Now, this is an experimental study where we compared individuals with subjective cognitive decline and controls, and we see the same difference that we see here with the meta-analysis. And now we're looking into, again, the electrophysiological patterns in these patients. And we also looked into imaging of different brain imaging. And so, for example, there's a study where we looked at the olfactory bulb size in individuals with mild cognitive impairment. There was no data about SCD. No study has been done. And we see here that individuals with mild cognitive impairment have a smaller olfactory bulb compared to healthy controls. Again, this is quite fascinating. And then with this, we did this study where we looked into the limbic system of the brain, which is the brain area that's responsible for memory, emotion, and so on, which is very early affected in Alzheimer's disease. And we compared healthy controls with mild cognitive impairment and subjective cognitive decline, but we subdivided these limbic brain areas into olfactory processing areas and non-olfactory processing areas based on previous studies. And what we see is that there is an impairment, a reduction of the volume in the MCI group of the limbic system, but only of the areas that are processing odors, not the areas that are outside the odor processing areas, but they still belong to the olfactory, to the limbic system. So this is, again, tells us there is some association between memory, olfactory function that we can see with imaging methods. We see it behaviorally. And so we think this is something that is also worthy to be looked at in more detail. So let me summarize very briefly. Parkinson's disease affects 1% of 60 years plus, Alzheimer's 5%. Both diseases are associated with olfactory dysfunction that precede the motor symptoms for Parkinson's and the cognitive symptoms for Alzheimer's. Now, with specifically to Parkinson's patients, we found a disease-specific pattern, and now we're trying to find better ways to assess this disease-specific pattern. So we have to do some method development. And this allows us to distinguish people that have lost their sense of smell, either because of Parkinson's or not. And that could, of course, be potentially useful for early screening. Now here I have a page where I thank people. So this is my team. And I actually noticed she's not with us anymore. He's not with us anymore. He just finished. She finished. So she just submitted her thesis 3 days ago. So I have to renew this. But this is the team, very nice team. I wrote this book in German for those who want to know more about the sense of smell. And these are the funding organizations that support our research. And of course, I would like to thank you first and foremost to listening to me. This is my email address. If you have any questions, well, we can discuss today, but we can also continue discussing in the future. Just reach out to me. Thank you very much.